Next up from our studio here in Stockholm, we have Modus Therapeutics, and presenting is CEO John Öhd. Welcome, John.
Thank you, Mike. And thank you everybody for having me and for your time. So let's talk about Modus Therapeutics and our trajectory towards developing a therapy to treat sepsis and septic shock. Modus Therapeutics is a small biotech company that develops its proprietary heparin-like molecule Sevuparin with a focus on sepsis and systemic inflammation. The unique aspect of Sevuparin is that it has been modified not to display the bleeding risk and the anticoagulation effect seen with the normal heparinoids. And this is what allows us to target higher doses that also can then explore effects in other important diseases that are not anticoagulation dependent. So what we have found in our preclinical models is that Sevuparin protects the blood vessels during septic inflammation. And in order to go further and realize development in patients with sepsis, Modus recently concluded an oversubscribed IPO on Nasdaq First North Growth Markets. The IPO proceeds are already at work to start our first study in this program, a phase 1b LPS challenge study. This is a sepsis mechanistic study, planned to start in Q4 this year or early Q1 next year. And this will then pave the way for the first patient study, a phase 2a study that is planned to start Q3, Q4 2022. So this is our team. It's a very seasoned team in terms of experience. We have representation from very experienced VC capitalists and big pharma executives. And then, of course, myself with a physician and clinical development background since long in different companies, and also our CFO, Claes Lindblad, with more than 20 years of experience from all aspects of the market and the economic control of life science companies. We also have a scientific advisory board that has been involved in the whole planning and setup of this new indication for Sevuparin, sepsis and septic shock. Sevuparin is a unique molecule that is based on heparin, but that has been targeted for modification chemically by modifying a couple of residues, 3 residues here. We go from the pentasaccharide that is typical for heparin and that gives the anticoagulation effects to a disaccharide repeat. This renders a patentability that gives us a patent that extends up to 2036, 2037 with extensions. And the modifications make it possible to dose Sevuparin in significantly higher doses compared to normal heparins. This is important because there's a broad set of mechanisms that can be targeted with relevance for other kinds of disorders than anticoagulation. And that is where our indication, sepsis and septic shock, comes in. So a number of different targets have been identified that are potently affected by a heparinoid, and we can target these in a much better way thanks to the modified molecular scaffold that constitutes Sevuparin. There is also another advantage. So we have finalized all the early development in terms of toxicology and phase 1 studies. We also have an extensive safety profile in man thanks to earlier development endeavors in sickle cell disease. So in all essence, we can then say that we are ready for phase 2 in sepsis and septic shock development. So looking a little bit closer on the mechanism of action that we have identified in this disorder, we have found that Sevuparin, the effects of Sevuparin helps protect the inside of the vessels during septic inflammation and systemic inflammation. And in brief, it means that normally in the bloodstream we have a calm situation where inflammatory cells are taking care of the monitoring of any immune threats, whilst at some occasions when bacteria comes into the bloodstream, this can lead to overactivation of these inflammatory cells. These activated cells then, in order to try to fight the threat that the bacteria is constituting, try to do so in some aspect by degranulating and releasing substances that are harmful for bacteria, but they are also harmful for the lining of the blood vessel. And this leads to a chain of events that eventually puts a patient to in a very poor spot, where organ failure ensues from the endothelia opening up and letting out plasma, so we have plasma leakage. What Sevuparin does, and what we have seen in our models, is that it can bind and neutralize many of these factors that are important for the damage to the endothelia in this early part of the reaction. So stopping the furthering of the worsening seen later. And just in brief, we can see that, for example, in the lung swelling in mice that are subjected to systemic inflammation, using Sevuparin, that lung swelling is taken back to normal, and also looking at cells that are endothelial cells. These cells open up on challenge by bacteria and white blood cells, but Sevuparin can put that back to a more normal state. And also here we have characterized the different targets that are constituted by these harmful agents and to what extent they are bound by Sevuparin and neutralized. Sepsis is a part of a bigger problem. It's the systemic inflammation disorders, so to say. And this is, just as with sepsis, this is where an inflammatory reaction becomes overdetermined and threatens the patient's well-being beyond what is clinically manageable often. And it can be caused by not only bacteria but also other big trauma, such as extensive surgery and extensive autoinflammation, autoimmunity. And as you can see, there are almost 17 million visits to the US ERs per year that go with some type of systemic inflammation, and sepsis is calculated somewhere between 2 and 3 million of these. And then, of course, the most severe form of sepsis, septic shock, is around 700,000 per year. And this is a very severe situation because the mortality is still around 30% here, and we don't have any specific treatments to use in this situation. So, starting out then with the clinical development to aim for this condition, we're planning to start in Q4 this year or early Q1 next year, a phase 1b study where we provoke a sepsis-like reaction in healthy volunteers. This allows us to study the effects of Sevuparin in this kind of systemic inflammation in a very controlled way, which will then serve to inform the important next study in septic patients. And this study then is planned to start Q3, Q4 2022, and to run for the subsequent year after that. This plan also coincides with our financing for the next study, for the phase 2A study. Right now we are working off the capital of the IPO, and within the IPO there were also warrants, and these warrants will vest and strike in a period that then is appropriate to finance the subsequent development. So looking at the treatment potential, if we follow the trajectory of a sepsis patient from coming into the ER, there are a number of different endpoints that we can study. We can study, for example, the symptom and the difference in symptom parameters between treated and untreated. We can also study the time to worsening or the time to recovery for patients. We can also look at the complications and death rate at day 28. So it's a fairly short treatment period, and also how long patients are in mechanical ventilation, which is a very important cost-containing endpoint. The development that we have discussed here is this part of development. Of course, later then there will be the need for phase 2B and phase 3 studies. And in our business model, we focus on partnering for this kind of indication. We think that brings value faster to our project. And for that, we need to present with potential inflection points. And we believe we have inflection points that drive value, both after the phase 1B study, but more significantly after the first patient study, and potentially we may need to also do a phase 2B study, after which there is of course also the potential for inflection and partnering. Modus is also continuously evaluating collaborations with value-enhancing potential. So a good example of that is, while not affecting our cost footprint so much, we're collaborating with Imperial College London on a severe malaria project in patients, and this whole project is financed by the Wellcome Foundation. So, to summarize, Modus Therapeutics is focused on its clinical program in sepsis and septic shock with its unique proprietary drug, Sevuparin. So we have preclinical data that indicate a protective role for Sevuparin on the vasculature during septic inflammation and systemic inflammation. We're phase 2 ready. The toxicology and safety packages are all in place, and we also have extensive safety data in patient groups and in man. This allows us a fast-to-market approach with clinical study already on track to start by the end of this year or early next year. And also this means that we have an early start to value development. Sepsis and septic shock is a leading cause of death in ERs and ICUs worldwide. And this means that with the market potential of around 6 million for septic shock market, that a successful treatment in this area would have a $1 billion commercial potential. We also have multiple shots on goal with further indications within the systemic inflammation area. As you remember, sepsis is a part of a bigger spectrum of disorders within systemic inflammation, and there could be possibilities, having a sepsis and septic shock treatment that works to also broaden into other types of systemic inflammations. We're also looking at patent protection up until 2036-37, and we're also conducting work that will have potential for new IP in pipeline. Thank you for listening. Thank you for your time.
Thank you so much for that presentation, John. I'd like to begin by asking you, in general, sepsis, what makes it so difficult to treat?
I think there's an easy and a difficult or a more complicated answer to that question. But I think there are a couple of basic factors. When we talk about treating sepsis, we actually talk about treating our own immune system, because that is what is driving the reaction the over-determined reaction of our immune system towards ourselves. So it's us fighting ourselves, because that makes it difficult. Another aspect is, of course, that it's a severe condition. So once we start treating it, patients may already be quite in a quite bad place, this means that, you know, this is related to another problem, and that is identifying the patients at an early stage enough to be able to have treatments that work. And the third step is, of course, also we don't have any specific treatments yet. So the treatments and standards of care that are used are general intensive care drugs. They are used in a good way, but I believe that in order to, you know, drive death numbers and complication rates down, we need to see more specifically approved medications coming onto the market and being employed at the correct time for these patients.
Absolutely. So, well, you just said that no other real treatments exist in that sense. How does Sevuparin compare to other treatments in development, let's say?
Yeah, so I think a unique part of what Sevuparin does is that we target a number of different harmful agents that participate in this reaction that harms the blood vessels from the inside. And I think we differ from most competition in that sense, because most other compounds in development have a more narrow scope, focusing on one molecule, one mediator, one cytokine of the septic reaction, or a specific organ, the lungs or the kidney. Whereas the effect of a heparinoid or a glycosaminoglycan here is to act in the whole systemic compartment, so to say, all of the blood, with this broad effect.
Okay, so much broader effect in general terms. So you mentioned the plan is to go to the clinic with Sevuparin. Could you tell us a little bit about how the clinical studies will be set up?
Yeah, so as I briefly showed in the presentation, we choose to start with a phase 1b study, which is not really a phase 1 study in its proper sense. Modus has concluded all the necessary regulatory phase 1 studies in order to go into patients. This special phase 1b study is a situation where we provoke a sepsis-like situation in healthy volunteers. That's why it's still called a phase 1b. So an LPS challenge study is where, in our case, we provoke both the skin very locally to simulate a local tissue inflammation, similar to what happens out in any tissue, with a small dose of LPS, with and without Sevuparin in different doses, and also where we give a larger quantity of LPS into the bloodstream of the patients, and then they really will experience a early reaction similar to the early phase of a septic or systemic inflammation. And then we also study the different doses of Sevuparin here. We hope that we will be able to extract very important observations here that will facilitate and make the next patient study more efficient and effective. Because in the real world, in the septic patient situation, things can be quite chaotic. And if we have important information such as dose level and indicators of when to start treatment and so on, that would be very valuable to trigger the patient study after.
Absolutely. Well, great. Well, thanks so much for answering the questions and for the nice schematic. It'll be helpful for our viewers for sure. And thanks for coming here today. We wish you all the best.
Thank you, Mike, and thank you everybody for your time and for having me here.